actual physically what you're doing today shouldn't take too much time. And as I mentioned in lecture, you don't actually turn these drawings in until the 8th of October, which is a week from today. The other thing you'll be turning in that day, and it's not on the syllabus itself, but it is mentioned in the study guide, is this handout that I just am assigning people. There are 5 different versions of them. They're all posted on Canvas. You're assigned to one of them by your last name's initial. So I've divided you into 5 different groups. So you can look at all of them though, but you only need to turn in one, and you're doing an analysis, a differential white blood cell analysis on that from the data that I put in there. So if we were in a real lab, you would be doing communally doing a differential white blood cell analysis. And what that would enable you to do is take a blood sample, a human blood sample. These are not something you're setting up yourself. You're getting commercial slides, since clearly handling blood is potentially dangerous. And you would have been counting 100 different white blood cells, and as you counted them, you would be categorizing them. This is a neutrophil, a monocyte, an eosinophil, and you would therefore have a chance to look at 100 different, you're going to look at a lot of neutrophils and a lot of lymphocytes and fewer eosinophils and basophils. So since we can't do that in person, then what I what I've done is I've taken the handout from our in-person class. I got rid of where you're adding your data. Instead, I inserted data, and these were abnormal data that I inserted, unlike in lab where you'd be looking at a normal slide. So your goals today are twofold. One is to draw the white blood cells. Now, unlike what I did with the cardiac muscle and the blood vessels and such, I don't have multiple versions of those white blood cells, but I don't identify on the histological slide what white blood cell is shown. So you have to look at it and determine what white blood cell it is. So let's talk about what they look like, so then that way you can help determine when you're counting them. Let me see, on the lab manual, there's a picture on page 420 in the lab manual. I'll write this on my shared screen. And these will give you some clues as you are doing this, how to tell whether it's a neutrophil, a monocyte, a lymphocyte, etc. What happened? Didn't make the actual connection, I guess. Okay, so neutrophils, remember, are the most common one. So most of the time people will not have a problem telling a neutrophil. So the key things about neutrophils and overall how you're going to tell these is by the size of the cell relative to a red blood cell. Some are bigger, some are smaller. And neutrophils are one of the medium-sized cells overall. I'd say you could have about 1 to 2 red blood cells would be able to fit inside of a neutrophil. They're not a really large cell. So just a little bit bigger, but sometimes because they may swell in preparing the slide, they could look quite a bit. They could look twice the size of a red blood cell around them. The real distinctive thing about neutrophils is they have multi-lobe nucleuses. And they're always more than 2 lobes, greater than 2 lobes. As soon as you see greater than 2 lobes, you know this is a neutrophil. Now I'm going to put this in parentheses. They are a granulocyte. The reason I'm putting a in parentheses is their granules are too small. If you looked really carefully, then you might be able to see these tiny, tiny granules or vesicles within the cell. The particular stain these slides are made with means to, the way it stains is it makes the granules generally looking purple or red, depending on the kind of substance that's in the granules. So in this case, it'd be purple granules. They'd be small and they'd be purple in color. The nucleus also looks purple. Most of these stains use, whether it's acid-loving or a base-loving substance. So basic items stain one color and acidic items stain another. So the nucleus stains purple as well, and the little granules stain purple. So often all you'll see is a slight purple haze color in neutrophils in their cytoplasm. Okay, those are neutrophils. If we were in person, it'd be really pretty easy to tell them from the other cells because of those multiple lobes right away. If it's more than 2 lobes, and there are regular lobes often, as you'll see in the pictures, it's pretty easy. Plus, you get to see so many of them since they're 40 to 70% of the white blood cells in your blood. So going in the order, the next one are lymphocytes. This is that never let monkeys eat bananas. These are the smallest, so they're approximately 1 red blood cell. Again, sometimes in preparation they swell up a bit, they look a little more bloated. That's probably abnormal, but they're the smallest for sure. And most of their volume is taken up by their nucleus. So very little cytoplasm is visible. So you couldn't tell that they are a granulocyte because you can't really see their cytoplasm is typically visible unless they get kind of swollen by the preparation artificially. And they're agranulocyte. So that, although I can't say that you can tell because you don't see much cytoplasm, it only has a very light purple color to it if you see it at all. The real distinction is the nucleus is a normal round nucleus, one round nucleus, and it takes up most of the cell. So that's typical of a lymphocyte. None of the other that you can see the nucleus have a round nucleus. Now, why I say that you can see them is that one of them, the basophils, it's very, very difficult to see the nucleus, so you really can't tell if it's round or it's lobed like the others. Okay. All right. Never let monkeys. So the monocytes. These are by far the largest cells, and they say we'll put 2 to 4 red blood cells, depending on how swollen they are. So these are the largest by far. You're going to see lots of cytoplasm. They're agranulocyte. Now you'll learn they do actually have granules. So this agranulocyte, it doesn't mean there really aren't any granules, it's just they're not visible, but not visible. So you will see a slight purple color to the, and actually this nucleus is purple, to the cytoplasm because they do have little granules. They are going to secrete quite a few things, but they're such tiny granules that they called them an agranulocyte back before they knew that they did have granules. So, so those are monocytes. So what about their nucleus? They tend to have a bilobed nucleus, but it's irregular. It's always got some kind of lobing to it, but it's irregular. Classically, you might say it looks like a dumbbell, 2 even-sized lobes, but sometimes the lobes look, instead of being, classically they'd be nice and round and they'd be bilobed like that, but sometimes they end up being kind of irregular shaped nuclei. They're still lobed, I didn't make that very clear. They're still lobed, but they could be kind of irregular. So probably the biggest hint it's a monocyte is its size. If it's really big and you see quite a bit of cytoplasm, and that cytoplasm doesn't have visible granules in it, then you're probably looking at a monocyte. You won't mix it up with a lymphocyte, even though they're both the agranulocyte, because you have the largest of them and the smallest of the white blood cells. Now, if you're not seeing them next to each other, you might say, well, how do I know? Well, that's why I'm giving you red blood cells as a measuring tool. Since all the red blood cells are similar sizes, you can compare your white blood cell to the size of the red blood cell. Any questions on that? Okay, you'd be struggling if we're really in lab to see eosinophils and basophils, since they only make up 2 to 4% for the eosinophils of all the white blood cells in your blood. These are highly distinctive. Eosin is a pink colored stain. This is saying they love eosin, and the reason they love eosin is because their granules stain pink in most stains. Actually, I don't remember in our slide if you have the classic stain with eosin staining. They have very large granules staining pink. You know what? I think the one we have is a right stain, and they're purple, stained pink. I believe we have the right stain. We have both of them in the lab, but I'm trying to remember which one I took a picture of. You're still going to be large, but they'll be purple. And they're very obvious granules. The others almost look like a little powder or something. Once you're on an eosinophil, they're very large, round granules you'll see, but they may be pink in some cases. Sorry, I did that again. Or they may be purple. Okay, well, basophils are going to have purple staining granules. So if you use the right stain, you're not going to be able to tell just by the granules. So you need to see the nucleus. The nucleus is always bilobed and it's symmetrical. Meaning you're going to see more like what I drew up there. You're going to see a little tiny bit of nucleus that's bridging two lobes like that. That's its nucleus. And you can see the nucleus. Even if they're both purple, the granules and the nucleus, you'll be able to see the nucleus. So those are eosinophils. What about size? These are fairly large. So they're not as big as a monocyte. So let's say they're 2 to 3 red blood cells in size. Again, depending on the swelling of the cells, abnormal swelling. So in the lab, when we're in person, if someone finds a basophil, then everybody celebrates and we all run over and look at their basophil. These are very distinctive. They're fairly large. Again, they're about 2 to 3 red blood cells, depending on how swollen they get. But the way you know is all you can see are the granules. There's so many granules in there, and they're so large that unless you use a specialized stain, you hardly can see the nucleus. So I'm going to just put large granules, obscure the nucleus. They're purple granules. And they're just chock full of these granules. They're little histamine bombs, basically, that once they start releasing those histamine granules, it's just a tremendous amount comes out, since they're just chock full of these. Yeah, they also secrete heparin, as does there are some other sources of heparin as well, as you'll soon learn. But mostly what they're containing is histamine. Okay, so these guys are so distinctive, I don't think you would have a hard time. So let's take a look at the pictures of them, and I think I stop sharing this now, and I'm going to share this one. I am going to go into the histology folder in Canvas and open up the white blood cells. So what you're going to draw today is, you see I have them numbered 1, 2, 3, and then this was just called 2 because there are 2 on there. So what you're going to do is open these and draw them and to try and determine what type of white blood cell that is. Now this is under oil 1000X. So I can't go any larger for this, than this. I can't blow it up anymore. And you might say, do I have to draw all these red blood cells? No, but I would at least draw some around because there are size markers, right? So yeah, so at least give some sense of the size by having some of the red blood cells. So you're going to draw each of those. The one that says 2 is because it has 2 in the view, even at oil. So these are all at oil. And then I made a mistake in the original writing of this. I thought I had 2 that were at 400, but I only have a 100 and a 400. So you don't really have to draw this one at all. I eliminated it over here in my version, and I'll put this version up so you have that. You're going to draw this though, and I'll show you the normal at 400, which granted, you can see at 400 what we get, maybe 4 white blood cells in a view. So even if we said, which is not really fair, but if we went from 400 to 100, maybe we'd have 20 or 30 white blood cells in a view. Does that seem fair? Hopefully. Yeah, good. So let's look at leukemia. And this is lymphoid leukemia, which means that the lymphoid line, one of those stem cells along it that generate lymphocytes, are making just a tremendous number of lymphocytes. And in fact, red blood cells are probably diminished in this, and other types of white blood cells are diminished. So for the lab test, all you have to be able to tell me is that this is a leukemia slide, and then remember if it's lymphoid leukemia. You can have myeloid leukemia, monocytic leukemia, lots of different kinds of leukemias. This is the type that's along that lymphoid lineage in the bone marrow. Okay, so those are the slides you're drawing. So that means you're turning in anywhere from 5, 6. If you want, you can draw that. I suppose I could show it just to show more, but it is something you already drew at 400. So 5 I'd say 5 minimal number of drawings, 1, 2, 3, 4, 5. Or you can, I wouldn't recommend combining this all in one drawing just because you tend to draw these really small. But, as long as you can recognize them on the test, that's fine by me. So that's what I have here. And as I said, I changed this. This said draw the 2 at 400, but I didn't have a 400. Now there's a tool to help you see some of the variations of these appearances of the different white blood cells that we use in labs. So I took a picture of it. So this entire row are neutrophils. So you can see in this photograph that neutrophils have multiple lobes. If I saw that one and I took a picture of it or I tried to do it in a differential white blood cell count, I would be struggling to identify that by the multiple lobes of the nucleus because the lobes aren't very clear. But you can see by the red blood cell next to it that it is barely bigger than a red blood cell. That means it's not an eosinophil or a monocyte.
Professor?
Yeah.
We don't see a picture right now.
Oh no, have I been going through this whole thing? You didn't see it?
Yeah, we saw like the
slide?
No, we saw the other things labeled like the white blood cell 1, white blood cell 2, that stuff, but we didn't see the picture of the actual.
Really? Okay, let me
Yeah.
go back and did I stop sharing? Let's stop sharing and share again then. Because I'm not sure why if I changed within my desktop, it would have shown something different. Okay, here's that poster. Can you see it now?
Yeah, we can.
Okay, I guess if I switch around even on my desktop, I've got to go out. All right, I just learned something. So here are neutrophils. You can see they're multi-lobe nucleus. They all almost look like they're not connected, but there are these little tiny nuclear bridges. There's really only one nucleus. It's just pinched off into these lobes. This one I was saying is the one that you might struggle and go, whoa, that looks kind of bilobed. Maybe it's a monocyte because the cytoplasm is mostly almost clear. It's not got big granules. Here are monocytes down here. But you can see the size is saying it's a small cell, not a monocyte. So I probably wouldn't put one like that on a test anyways, and I didn't take a picture of that. They're all going to have multiple lobes like this guy if it's a neutrophil. So never let. Is there something else, Dina?
Oh, no, I was just going to say if you take your PowerPoint and like you're going to run the whole PowerPoint, it'll enlarge it for us so we can see it a little better.
Okay, I was going to switch. All right, I got to unshare to do that. I was going to switch slides.
You just start.
Yeah, I can do that. That's a good idea. Okay. Play from current. All right, now I have to go. Wait a second. I can't get to you guys if I do that way. Let's see if it'll let me do it that way. Of course, I must be able to do that, right?
Professor, I had a quick question. Do we have these slides available on Canvas yet? Because I'm not seeing it.
They are. They're in this lab test 2. Here we are. So they're the study guide, the PowerPoint under the folder that says Lab Exam 2. And then probably near the bottom is the PowerPoint. Once you open that folder. Are you finding it now, Noah?
No, the only PowerPoint on here is the spirometry lab. And that's in the lab test 2 materials.
Okay, let me exit this and see if for some reason I get another Chrome window here. Mine has it and yours doesn't. I don't, because I was in it this morning looking. That's why I was confident that you guys had it, but maybe I was crazy.
Yeah, I don't see it anywhere.
Lab test 2 materials. You're right, it's not there. Okay, let me upload it. I guess I was looking at it in my files. Where is it? I'm on desktop. And there we go. Lecture slides. Should be under, oh, there it is. Okay. All right. I'll upload it, which will take a little bit since it is live assignment 2 study guide. Take a little bit since it's a PowerPoint, which is a little bit large. So these are the only 2 pictures I'm going to show you. While that's doing its thing, which is going very slowly, let me go back to sharing this slide. Then you'll have it when you go to do yours. Dang, where are you guys? Too many windows again. There you are. Okay. File. This one. All right, let me do the thing that, okay, are you seeing this poster now? Again?
Yeah, we are.
Okay, great. All right, let's go down and, oops, dang. And look at the monocytes down here. So, well, lymphocytes first, never. We have neutrophils. See, lymphocytes very tiny. They're about the size of a red blood cell, but if they get a little bloated, they might be too. This would be an abnormally large one. This one is probably, about ready to burst. It's got some sort of osmotic problem from being put on the slide. These two here are far more typical. So I took a picture, as you saw a moment ago, similar to these two. So measure them up against a red blood cell, since the vast majority are going to look like this or maybe like this. You're not going to see lobes at all in the nucleus. That would be very unusual, but they are showing you the whole range here. So here are monocytes. They're huge. You can get 3, maybe 4 or 5 red blood cells inside of one of these. Their nucleus is bilobed, but it's highly irregular. This looks, I'm sorry about the reflection on the poster, but this looks kind of like 2 even-sized lobes or a little bit of an indentation lobe, but then you get these big sprawling things. Their cytoplasm is really, really clear. They are big, big cells compared to pretty much anybody else here. You can see basophils and eosinophils are fairly large too, but not as big as the monocytes, which are extending from here to there. Okay, the last two pretty easy. Here are eosinophils with the pink stain in their granules. Their granules are so big they look like little those drinks that you drink with. I'm trying to think of what the little bubbles are in there. Gosh, my brain just blanked out on what I'm thinking of, but they've got little granules that are very clear inside here. And bilobed nuclei, these are both bilobed. These are eosinophils. Now, I think in ours, although we could check, these might be purple, but you'll still see the bilobed nuclei. And that's opposed to the basophils. There's so many granules. There is a nucleus. I don't actually know what its shape is, but sometimes you see a little peeking of it amongst all these granules, but it's really hard. There's so many, they're so dark staining, and they're so large that they just obscure the nucleus altogether. All right, so you should be able to see all of this in a minute. Let me see how my uploading is doing. I'm gonna stop sharing and go over and see how that is faring. Oh God, so many freaking windows. Yeah, it's still uploading. It's probably because I'm doing this on the internet that it will upload by the time you get to it. So you do have the white blood cells drawings. So that should be in your folder, right? That's already there. Am I good on that? Anyone just speak up? You can say yes.
Yes.
Yeah, all right. The other thing that I added this morning in that folder, I'm going to show it to you, is the differential white blood cell count handouts. Okay, so I'll share this screen. So this one, there's 2 of them up here. Okay, do you see this folder here? Am I sharing it okay?
Yes, we can see.
Okay. So if you open this folder, there are groups, and the groups, if you look at the full name of this, which is group 1. So group 1 is going to be for people whose last name begins with A or B. And you'll see that I've entered what would be like a data that you may have collected on a patient's white blood sample. So you've counted 100 cells. That's why they're all out of 100 white blood cells, not 100 neutrophils, but 100 white blood cells. And you found that about 50 of them you identified as neutrophils, and then et cetera for the rest. So what you want to do is figure out which one of these is abnormal. These are all going to be abnormal. Remembering that abnormal low is because potentially something else is high. Since you're only limited to counting 100 cells. If one cell type goes up in number, the other has to apparently go down in number. So look at this and then determine which of these conditions, know what causes these conditions. You're going to study this whole column. And then you're just filling out this part. Tell me what condition this one has. And then 2 reasons out of the possible, there could be more than 2 reasons here. Is that clear?
Should we be seeing the differential WC count exercise opened up?
I didn't understand you, Dina. What did you say?
Should we be seeing the actual folder opened up?
Oh, you didn't get that?
No, ma'am.
Sorry. Guess I can't open a folder without my perspective and your perspective. Okay, let me close that. I'll open the folder and then I'll do another share. Okay, now it's taking a long time to do this. All right, so I guess I have to go out and share again. It's still taking a long time. Okay. All right. This thing is awkward. Okay, here it is. Yeah, it just keeps adding new windows. Sorry about that. Yeah, just speak up sooner and when we get through this. But I'm showing you here 100 cells were counted. 50 of them were neutrophils, et cetera, down there. And then at the very bottom, you're just telling me what condition does this particular data suggest this patient has, and 2 reasons. Now, where you find that is over here in this column that you're going to learn. Okay, again, all of this on the second lab exam, not this lab exam. All right. Are you guys good? So notice one thing I said, don't put leukemia as an answer. There's more, I mean, leukemia is, even if you were more specific and you said it was lymphoid leukemia or something, maybe, but I'd just rather you look at some other conditions.
Is one of my biggest fans.
Whoops, did you have a question, Adrian? No, okay. Because leukemia, there's so many different types. And whenever you see something elevated, the temptation, in my experience with students, is to call it leukemia and just let it go at that. So I'd rather you not choose leukemia as these. You should be able to come up with some other answer. This is also defined in the lab manual. Leukocytosis is a condition where you don't have just one cell type elevated, all white blood cell types are elevated. So at any rate, the leukocytosis, I would know the definition of. Leukemia, know the definition of, and understand how different lineages give you different types of leukemia. So that's the activities for today. So if you can, remember you're turning this in 1 week. If you're not sure which group you're in, then go into the announcements. I'm going to stop sharing. Go into the announcements, and I've assigned it by the letter of your last name, not your first name. So, so go ahead and just fill out that one. But if you just curious, eventually we'll go through this handout, probably the day it's due, and we'll talk about which, we'll do all 5 of them. What are the conditions in all 5 of them? Are we good? Are there any questions? Looks like there's a whole bunch of chats. So there's probably all about not there. You don't see it. Yeah, so most of it's about that. I'll look and see how the loading is going for that. It's almost done for loading up that one in Canvas. And so you should have that available, but I don't think it's critical since there's only 5 drawings. You could probably figure out from the ones I took pictures of what they are. And you have pictures in your lab manual on page 420. So there's a single picture of each type on the lab manual. So if you don't have the poster, you should be fine. Okay, any questions? So if I turn you loose, let's see, how do we know which group? You go into announcements on Canvas. And in the announcements that I put up this morning, I told you which group, but it's by your last name. So since your last name is C, you're probably in group 2. I know group 1 is last names beginning A and B. So you're probably in group 2. Okay. All right. So just give me a shout since I may be going around between other screens and having lunch or whatever. So if you need any help, and if you'd like to be put into a breakout room, I'm happy to do that too. Okay. All right, I'm going to mute myself and let you guys get to work.